Hi everyone. Okay, we're just going to spend the first few minutes making sure you're all in position, but welcome to another free online class. Can you hear me? Yeah, perfect. Hello everybody. Welcome to today's online classroom on digestion and enzymes, which is one of my favorite topics. So hi, whoever's from Malaysia, how exciting. Okay, so when we talk about digestion, it's all about how we take the food that we're eating at least 3 times a day, I imagine, and how we get it into a form which means that it can be absorbed into our bloodstream. So let's get your piece of paper and your pen ready. As always, I've got some questions to ask you so you can check your understanding too. But we're going to start by looking at our definition of digestion. So it's the breaking down of large insoluble food molecules into small soluble ones, and that's so that they can be absorbed into the bloodstream. Crucially, the insoluble food molecules, they can't move into our blood until they've been converted into a small soluble form. So that definition is definitely worth writing down. Okay, so as we can see below that, I've broken down digestion as to being one of two types. There's both mechanical and chemical digestion. So with mechanical digestion, I want you to think about how that's just the physical breaking down of food. It's the process of taking large amounts of food and breaking them down into smaller pieces. So if you've got a lump of food here and you literally just chop it up into smaller pieces, and by doing that, I'm hoping you can see, but you're increasing the surface area, which is really important. We really want to increase the surface area of that food. And you'll notice that when you chew your food, that is what is taking place in your mouth. You're taking that large portion of food and you're breaking it down. Notice you are not changing the structure. We're not inherently changing that food and how it's composed. We're just breaking it up into smaller pieces which have a larger surface area. If we look at chemical digestion now, this is very different. This is actually taking food molecules, and what you're doing is you're actually changing the structure of them. So for example, if we take a large food molecule such as starch, when it undergoes chemical digestion, you don't just have smaller pieces of starch, you have a totally different molecule, which is smaller and is known as maltose. So we've gone from having the insoluble starch and we've converted it into the smaller soluble maltose. So hopefully you can see that through both mechanical and chemical digestion, together you're going to end up with your definition of digestion, which is how you break down your large insoluble food molecules into small soluble ones. So it's definitely worth bearing in mind that there are two types of digestion. Yeah, sorry. And when we look at chemical digestion, I'm hoping that some of you are already aware that really what we're looking at here is enzymes. Enzymes are totally key for chemical digestion. Yeah, it will be on YouTube later, Edward, so don't worry about missing the beginning. Okay, so enzymes now. I've told you that they are essential for chemical digestion. Notice that what they do is they speed up reactions without being used up. So that's why they're so useful, because you can use them repeatedly. They're also highly specific, which means they only work on certain substances. And we're going to look at the examples of enzymes you need to know about in the digestion topic. So let's have a look at some examples of enzymes. Here we have our starch molecule. It's a large insoluble molecule. And I've already told you that it gets broken down into a small soluble molecule known as maltose. However, we need to know which enzyme is responsible for this, and it's actually an enzyme called amylase. The enzyme amylase is very specific and it will only work on starch. They're quite fussy about you knowing where the amylase is actually made and where it acts. So I'm just going to make a little note here. The amylase is produced in the salivary glands, the pancreas, and the small intestine. Maltose and glucose are both sugars. They're small sugars, and it depends on your exam board. They're kind of used interchangeably, but if you want to be an accurate biologist, you need to be aware that they're different sugars. So the amylase acts in both the mouth and the small intestine. Yeah, exactly, Zara. Now, a second type of digestive enzyme is protease. This is nice and straightforward in that the name is similar to what it acts upon. So this time our protease is going to act on a different nutrient. It's going to act on proteins, and it's going to help break those proteins down into small subunits known as amino acids. In terms of where the protease is made, it's made in the pancreas, stomach, and small intestine. And it acts within the stomach and the small intestine. Okay, and then I was making some notes to myself here. The last enzyme we need to talk about is the enzyme lipase, which acts on fats or lipids, if you're feeling fancy. It breaks down the fats or the lipids, excuse me, into fatty acids and glycerol, so slightly more complicated. Now, lipase is made in the pancreas, and it's acting in the small intestine. One thing I wanted to point out is that enzymes are made out of protein, and it means that they get damaged at high temperatures. So they're quite fussy proteins. They like a very particular temperature range, and we call that permanent damage. We say that it's been denatured. And there's a lot more to say about enzymes, but I'm really keen to make sure we go through all the different parts of the digestive system, so I won't talk about those here. I just want to make it clear that enzymes are denatured at high temperatures. Okay, let me know when you're ready and we'll move on. Okay, perfect. So here we have a diagram of the digestive system, and it's quite important that you guys are aware of what goes on in each part of it. So starting with the mouth, obviously that's where we take in our food. This is known as ingestion. And what's actually going to take place in the mouth? Well, hopefully you can hazard a guess that because we have our teeth in our mouth, we know some form of mechanical digestion will take place as the teeth break up the food. Why? To increase the surface area. So you can write some really nice detailed answers here. In terms of what's going on in our salivary glands, which are also present in our mouth, well, remember that the salivary glands are releasing the enzyme amylase, so we know that starch is being broken down into the sugar maltose, and that's why if you chew a piece of bread for long enough in your mouth, it will start tasting quite sweet, and that's because the starch in it's been converted to the sugar maltose. Next up, the esophagus, otherwise known as the food pipe. You can spell that with an O, by the way. This is quite an American spelling here. Not much to say here. We can say that the process of peristalsis takes place in the esophagus, which is just the contraction of muscles which help force the food down. Okay, it just totally depends on your exam board, but yeah, maltase does convert maltose into glucose. Okay, next up in the stomach, which is this sac. And obviously the stomach is full of an acid. This is hydrochloric acid. Now that hydrochloric acid helps to break down the food further, but really its primary role is to kill bacteria. And that's going to stop us getting sick if we eat some food that has bacteria on it. If obviously we take in rotten food or really nasty food that's got too much bacteria, then that's why we get sick. Hopefully you also remember that the stomach produces the enzyme protease, which, remember, carries out the chemical digestion of proteins into amino acid. Okay, moving on to the liver now, which is one of the largest organs in the body. So the liver produces bile. Now, bile is a super important substance because it has two roles. It emulsifies. So what does emulsify mean, or emulsification? Well, it means that it breaks down large fat droplets into smaller fat droplets. And this is really important because it increases the surface area of those fat droplets. And if you remember from above, which enzyme is then going to work on those fats? Well, it will be the enzyme lipase. So effectively makes it easier for the lipase to get to work. So the first role of bile is to emulsify, so breaking up large fat droplets into smaller ones. The second role of bile is that it neutralizes stomach acid. So it's quite an important substance. You also need to be aware that the bile is stored in this little green object known as the gallbladder, and that bile is released into the small intestine. So that's actually where it gets to work. So a lot of exam boards are very keen in asking you, number one, where bile is made, number two, where it is stored, and number three, where it gets released into. Bile is made in the liver, stored in the gallbladder, released into the small intestine. Okay, moving on now, the small intestine. This is really important because after all this work we've done on our food, it's now ready to be absorbed. So it absorbs small soluble products of digestion into the blood. The large intestine absorbs water, and really, we're nearly at the end of the digestive system now. All there really is to add is feces. So feces is the scientific word for poo. The rectum stores feces. And then the anus is just a ring of muscle known as the sphincter, which allows feces to pass out of the body. And if you're feeling fancy, you can call that digestion. Jennifer, in terms of absorption, at GCSE, they'll just ask you about diffusion. At A level and IB, they'll want you to talk about every single nutrient and how it's absorbed, whether it's by active transport or diffusion or protein carriers. It's never going to be osmosis. Yeah, Zara. Please let me know when you want to, I've written it on the iPad. Digestion is the removal of feces from the body. So pooing, really. No, that's not right about the active transport and the small molecules. It totally is dependent on the food molecule you're talking about and whether it's hydrophobic or hydrophilic. But again, that's what you study when you're 17 and 18. Okay, let me know when we're ready to move on and look at the villus. Great. Okay, so hopefully you're aware that your small intestine is a very, very long structure. It's like a very long tube that the digested food products pass along, and then effectively those digested food products get absorbed through the wall of the small intestine and they enter the bloodstream. So I'm hoping you can see the top diagram is a tube. That's where all those food products are going to be. And the weird little projections are known as villi. Now, these are so important because if we remember, what happens in the small intestine is that absorption of digested food products occurs. And so really, we need to work out how we can make this process as efficient as possible. And that's what this little diagram is at the bottom. You can think of it a bit like when you dry yourself with a towel. Think about the structure of a towel. It has all those little bumps on it. Why? To absorb as much water from your body as possible. How? By increasing the surface area over which that absorption takes place. Absorption within the small intestine is pretty much the same. So they love asking you about adaptations of the small intestine for absorption. So number one, we're going to say the presence of villi increases the area for absorption. There's also microvilli too, but this is enough for at least 1 to 2 marks. What else can we say? Well, we can see this little red line here. This is the presence of blood capillaries. So many blood capillaries ensure absorption takes place quickly. In terms of distances involved, we're talking very short distances. So we say short diffusion distance. So at GCSE and IGCSE, we're assuming that kind of incorrectly that everything is moving by the process of diffusion. And diffusion is definitely a topic for a different video, but it's just the way in which substances move from a high amount of the substance to a lower amount of the substance. And then another thing to mention, if you're feeling fancy, is the presence of lacteals, which enable fats to be absorbed. And this is going to get you 5 marks in an exam. As long as you use these words, you're nice and technical. And as always, let me know when you're ready. Okay, great. Now it's over to you guys. So here's a question. Now I want you to write down the letters A to J, I think, and just on your piece of paper, obviously don't ruin it for everyone by putting it in the group chat, and just make sure you can label all parts of the anatomy of the digestive system. Oh my goodness, Anna, I don't know. I really don't know. I've never seen anything like this before. I don't know if it'll be September. I know schools don't do that much teaching in the final term. I don't know. It's also like people that pay for school fees. Are they paying full amount? So many questions I don't know the answer to. Okay, so let's whiz through these. A is the salivary gland. B is the stomach. C is the large intestine. D is the rectum. E is the anus. F is the small intestine. H is the liver. I is the esophagus. And J is the mouth. Did everyone get those all right? Oh man, I always do this. This is why Martin's here. Well done, everyone. Okay, more questions for you.
Scroll up the comments, see if you've seen Edwards about private school.
Yeah, I did. What did he say? I go to a private school and they're offering fee reductions 10%, 20%, 25%. Presumably boarding schools have to offer a huge reduction seeing as people aren't sleeping or eating now. Oh wow, Shanghai's getting ready. Yeah, Edward, I assume you're British, but yeah, some schools have lots of Chinese international students. Okay, where's amylase made? That is A and G. So the salivary glands and the pancreas. Some of you might have put small intestine. I don't have a problem with you putting that. Where are the feces stored? Well, remember that was in the rectum, which is D. And then next up, where is the protein digested? Well, that occurs in the stomach, which is B, and the small intestine, which is F. So in this question, they're really making sure that not only do you know the anatomy of the digestive system, but they're also making sure you know all the various roles. So although it looks fairly straightforward, all you're doing is listing letters. It requires quite a lot of knowledge in order to do well in this question. Did everyone get those right? Yeah, well done, everybody. Okay, and this is exactly the sort of question I'm talking about, worth 5 whole marks. Describe and explain how the structure of the small intestine is adapted for absorbing digested food. I wouldn't recommend writing an actual bullet point, but what I say to the students I teach is to effectively write bullet points which run into each other. So it won't be the best sentences you've ever seen, but you could write something like, the villi increases the surface area for absorption, full stop, blah, 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 does blah, blah. So it's effectively bullet points without drawing them. I wouldn't personally recommend writing bullet points like your teacher said. Okay, let's just go through the answers. So 1 mark is available for saying that villi increase the surface area. 1 mark is available for saying there's a good blood supply or good capillarization in order to absorb the nutrients. There's a mark for saying that there are thin walls or a short diffusion distance. You can use lots of different ways of saying the same thing. Martin's going to type up the perfect answer for you, by the way. There's also a mark for saying that lacteals are present, which absorb fat. And so you can write a fairly concise answer by not writing loads and loads. You don't want to write 20 lines for this. You just want to make sure that what you're writing is very heavy on scientific content. Okay, guys, right, we're going to have to stop there, mainly because we're running out of time, but we're going to have to try harder at not ruining it because honestly, you're all out of control. Okay, I'll catch you soon, and I hope you found this lesson really helpful. Right, take care, guys. Bye.